I'm Kavya from First MSc Botany. Here we are going to see about life cycle of Equisetum. Habitat. The plant body of an Equisetum has an aerial part and an underground rhizome part. It is commonly known as horse tail or scoring brush. Majority of the species are small with a size range between 15 and 60 cm in height and 2 cm in diameter. Generally, it grows in wet or damp habitats. Some species adapted to xeri condition. Example, Equisetum urbanis. Structure of Equisetum. The sporophytic plant body differentiated into stem, root, leaf. Stem. The stem of Equisetum has two parts: perennial underground much branched rhizome and an erect, usually annual aerial shoots. The branching is monopodial. Shoots are differentiated into nodes and internodes. Some shoots are profusely branched, green and purely vegetative. Others are fertile, brownish and have terminal strobili. The internal structure of stem. The stem of Equisetum appears wavy in outline with ridges and furrows. The epidermis wall is thick, cuticularized and have deposition of siliceous material. Stomata are distributed only in furrows. A hypodermal sclerenchymatous zone is present between each ridge. Cortex is differentiated into outer and inner. The outer cortex is chlorenchymatous while the inner cortex is thin-walled parenchymatous cells. There is a large cavity in inner cortex is corresponding to each furrow and alternating to each ridge. It is known as vallicular canal. The vascular bundle are arranged in a ring which lies opposite to ridges and alternate with vallicular canal of the cortex. Vascular bundles are conjoined, collateral and closed. In the mature vascular bundle is disorganized to form a carinal cavity which lies opposite to each other. The vascular bundles remain unbranched until they reach the level of node. At the nodal region, each vascular bundle trifurcates, which means it divides into three parts. Root. The primary root is epimeral. The slender adventitious root arises endogenously at the nodes of the stem. In tears, the root shows epidermis, cortex and stele from periphery to the center. The periphery consists of elongated cells with or without hairs. The cortex is extensive. Cells of the cortex often have thick walls. Sclerenchymatous and those of the inner cortex are thinner parenchymatous. A large metaxylem element is present in the center of the stele and the protoxylem strands lie around it. The space between the protoxylem is filled with phloem and there is no pith. Leaf. The leaf of Equisetum has small, simple, scale-like and isophilous. They are attached at each node, united at least for a part of the length and thus form a sheath around the stem. The sheath is free and pointed teeth-like tips. The number of leaves per node varies according to the species. The species with narrow stem have few leaves. Example, Equisetum scripvoides, and those with thick stem have many leaves. Equisetum scaphinare. The number of leaves at a node correspond to the number of ridges on the internode below. The leaves do not perform any photosynthetic function and their main function is to provide protection to young buds at the node. Antheridium. In monoecious species, Antheridia develop later than Archegonia. They are of two types, projecting type and embedded type. Antheridia first appear on the lobes of gametophyte. The perennial division of the superficial antheridial initial give rise to jacket initial and an antragonial cell. The jacket initial divides anticlinally to form a single layer jacket. The repeated division of antragonial cells form numerous cells which on metamorphosis produce anthrozoids. The anthrozoids escape through a pore created by suppression of the apical jacket cell. The apical part of the anthrozoid is spirally coiled, whereas the lower part is to some extent expanded. Archegonium. Any superficial cell in the marginal meristem acts as archegonial initial, which undergoes periclinal division to form a primary cover cell and an inner central cell. Cover cell by two vertical division at right angle to each other form a neck. The central cell divides transversely to form a primary neck canal cell and a central cell. Two neck canal cells are produced from the primary neck canal cell, while the ventral cell by a transverse division form the ventral canal cell and an egg. At maturity, an archegonium has a projecting neck comprising of 3 to 4 tiers of neck cells arranged in 4 rows. two neck canal cells of unequal size, a ventral canal cells and a neck at the base of the embedded center. Fertilization. Water is essential for fertilization. The gametophyte must be covered with a thin layer of water in which the motile anthrozooids swim to the archegonia. The neck canal cells and the ventral canal cells of the archegonia disintegrate to form a passage for the entry of anthrozooids. Many anthrozooids pass through the canal of the archegonium, but only one of them fuses with the egg. The diploid zygote is formed. Generally, more than one archegonia are fertilized in a prothallus. Embryo. The embryo is the mother cell of the next sporophytic generation. Unlike most pteridophytes, several sporophytes develop on the same prothallus. The first division of the zygote is transverse. This results in an upper epibasal cell and lower hypobasal cell. The embryo is therefore exoscopic. The epibasal and hypobasal cells divide at right angle to the oogonial wall, and as a result of four cell quadrant stages established. All the four cells are differentiated. The four cells embryo undergoes subsequent division, and the future shoot apex originates from the larger cell and leaf initial from the remaining cells of one quadrant of the epibasal hemisphere. Later root grows directly downward and penetrate the gametophytic tissue to reach the soil. A number of sporophytes may develop from a large nature gametophyte if more than one egg is fertilizer. And it is the life cycle of Equisetum. Thank you.